So I just could not resist this picture showing the walking with caveman, and really how that does a great job demonstrating human evolution. But getting into this last topic, our central idea is that meiosis leads to this idea called independent assortment. And this is something that Gregor Mendel, oops, misspelled his name, came up with, saying basically genes all get sorted, all move around independently, and get, like a deck of cards, get randomly distributed when gametes are made by sexually reproducing organisms. Okay, so basically the idea that genes are randomly, we'll say, mixed up and distributed Remember, gametes, how we normally think about it, we think of sperm and eggs, at least for humans. And then those go on to, if we have fertilization, we have it, and we can see these unique patterns when we look at the offspring. And something also, Mendel was a huge influence, but also this other scientist called Thomas Hunt Morgan, because he and others noticed that the Mendelian ratios, the patterns that Mendel laid out, didn't always hold up. So noticed that those patterns we'll say hold up. There were other ideas that needed to come to the fore to account for what was happening in Thomas Hunt Morgan. And he's the fruit fly guy. Also a real classic biology experiment. Really one of the most classic ones is growing fruit flies. I did that when I was in high school, but it's not done as much now. There's some other methods we have to see though. But maybe I should do that. Maybe I should do some fruit flies. That might be kind of fun, but pretty tricky to get all the mating done right and counting them. But fun in its own way. So it's really important whenever we see anomalous data, anomaly, out of the normal, of the expected, out of the normal or expected, we pay attention to that. Doesn't mean it means you're discovering a new law every time something goes wrong. Things go wrong all the time when we do labs. It doesn't mean we're discovering new concepts out of the norm. But we take into account when we really explore those things. Expected. So that's just the nature of science. We always have to be pushing forward using the best available evidence that we have. All right, so before any of this happens, meiosis or mitosis or anything, you need to have replication. So this is the normal existence for our chromosomes. Normal, they're just hanging out, but then they get replicated in the S phase. Copied. And you can see that here, okay? Each of these has grown a new half, right? And now we see them like this. Remember, this is a copy of the other side. So they're exactly the same. Then in meiosis 1, these copied, these homologous chromosomes that have been copied are separated. And this is when, like right here is 2n, this is 2n. When this happens, it goes into n, so it's half the number. So if this were a human, it'd be 46. 46, we're all good still. And right here, we're looking at 23. And then we have meiosis 2. You see here at meiosis 2, these copied chromosomes are separated. Remember, these are identical. Identical. These are the same. Exact copies, these two pinks and these two orange ones. All right. This side here kind of looks like mitosis because you're just separating those copied chromosomes, which is what you see in mitosis. But you got to have that replication first. Once you have that replication, then you can go through meiosis. One of the most important things about meiosis is this crossing over, this exchange of information between non-sisters. So these two chromatids here are sister. But if we go here and here, these would be non-sister. And this kind of area where they come together, that's called a centromere. So we can kind of see the two sister, and then these two are non-sister. We can see them kind of binding together, you see here, and then right about here. When they're bonded together in that kind of X pattern, it's called the chiasma. And those two homologous chromosomes, that name for that is tetrad. Also, you'll see the word bivalent. So it's kind of confusing. So, sometimes with the chiasma, they will actually switch sides. So you see over here, the capital is on the right, the lowercase is on the left. Here, they switch, right? They transfer. So the segments of non-sister chromatids are exchanged. And then when you have here, these two, see these two alleles here and here, they're a little different than they were originally. Okay, and these are called our recombinants. We'll draw that out and show that. And then next we got some more. I got some more slides to go over that stuff. Here we go. So the new combinations, and we can see them out. You'll see the alleles drawn this way. This is one allele in the pair. This is the other. It's been copied, right? So you see it copied. So it's capital E, lowercase, lowercase d, uppercase E, right there. But then we can see this will get exchanged there, and this will get exchanged here. And you can see the recombinants right there. So just be able, when you see a line under here, just know that's an allele. And you'll see in the parents, we did not see a capital D and an E together, did we? Like right here, we didn't see the capital D and the E together on an allele. But now we have that. So what do you have? You have more variation. So this crossing over leads to more variation. Okay, and as it says here, standard notation for genotypes. So just, on the IB exam, they'll probably use that notation, or they will use that notation. So more about these chiasma. Remember, these are homologous chromosomes. Remember, they're the same shape, size. They got the same kinds of genes. Like you have the eye color gene over here, say this is the eye color gene here on this one. This, you're going to have the eye color gene over here on that one. And you can also look at it this way. This was from the mom, and this one is from the dad. You can look at it like that too. Now, when they're coming together, if you remember, the term, it's either bivalent or it's called a tetrad. And this is where you have that switching. And you'll see here the recombinants, kind of like I labeled earlier. Okay, you have that gene, these genes exchange. Actually, it's the other way there. So, the blue went that way, and the pink went that way. And you have basically new genetic material. So this allows you to adapt to conditions better, allows you to survive if the conditions change. So the main thing to know in meiosis, the first step, the homologous chromosomes are separated, and this is where you go from, in humans, 46 to 23. You can see the homologous pairs here lining up at metaphase 1. And they're kind of linked together, right, where they may have switched. And we see they did switch there. Boom, boom. Okay, we have recombination. And remember the phase where they pull apart, that's anaphase. Anaphase 1, they're pulling apart. Now remember, once this happens, they're not being pulled, they kind of they walk each other, they walk themselves along these microtubules. So once we're here, we're moving towards the end, from 2N to N. Just gotta remind yourself of that. And in meiosis 2, you have the sister chromatids separate. Now these normally are copies, right? Normally exact copies. But look here, are they exact copies anymore? No, they're not. So they're not copies anymore. That's if there was recombination. So now we're going to have those sister chromatids are going to become separated. So this is metaphase 2. And then anaphase 2. And that, remember, they're right now they're N. I'm sorry, yeah, they're single N, 23 for humans. When they separate, they're still going to be 23 because remember, these genes are copies of each other, so they're just kind of redundant information. So you're not really changing the amount. You're changing the amount of DNA and genes, but not the information. You still have the same. It's like separating two copies of a book, right? Each side still has the same information. So that is the second part of meiosis, where those sister chromatids are separated. And it all has to do with how the pairs are oriented. Now this is one I'm going to write down possibility 1. This is possibility 2. And you'll see here, we'll say the blues, I will just be gender stereotyping, I guess, and say those are from the dad, and the red one is from the mom. And this one, say there's only for this organism, there's only 2 chromosomes. So, chromosome 1 and chromosome 2, and not, 23 pairs. So just 2 pairs of chromosome for this organism. So the chromosome pair from the dad lined up on the left and the mom lined up on the right. And you can see if that happens, you can see how the separation works. We have these ones are all from the one parent, these are all from the other. And then you get these are the possibilities you're going to see in the gametes. Now over here, you'll see they lined up slightly differently, right? Just by random chance before they were separated. Like this one, say, switched. And because it did that, there's a different outcome of the gametes. You see how there's different combination? These are different combinations. All based on how they were oriented before they pulled apart. So, two different chromosome combinations. Now, what you need to do is draw that. So, let's go ahead and do that. So, we'll say I'll keep, let me change this to green. Yeah, that's a little bit lighter. So we'll say that's one of my chromosomes. You know, it's been copied. Like a big old H. And here's the other chromosome. Oh, that's kind of ugly. Anyway, this one is from. Oops, this one is from the mom. Say this one is from the dad. Now we're going to move on. We're going to kind of blend those together. Should have drawn that out a little bit. Oh, the whole thing. There we go. This one's kind of going over like that. Not going to be too symmetric. And see, we're going to have some switch outs here. So let's. It's going to now have that little bit of red on there. If your drawings aren't as good as mine, don't feel bad. You know, it's the best you can. And this side, there we go. Now we'll make that green. I could hold on, let me. Yeah, so that's some crossing over. Okay, and these are my recombinants. These two, let me write it. These two alleles right here, these are my recombinants. Okay. And maybe let me go back. I should have, we'll say this was a capital A right here, and this was a little a. Over here, we would now have this little a over here and this capital A over there. Switch that allele. So that is 10.1.